Alright, hey guys, welcome to 8.2. In this video, we're going to look at reproductive cloning. Let's jump into it. Okay, so cloning is the artificial production of new individuals with the same genetic material as the parent organism. You can think of it as a form of asexual reproduction, but it's an artificial process done by humans, or at least as far as we know, it is done by us. Who knows, maybe there are animals out there that are also artificially cloning each other. So basically there are four processes of reproductive cloning that we're going to cover in this video. The first one is looking at cuttings and graftings. We're going to look at tissue culture, embryo splitting, and somatic cell nuclear transfer, or simply known as nuclear transfer. Okay, and then here's just each of the examples there. So this is cuttings and graftings, this is tissue culture, this is embryo splitting, and this is looking at somatic cell nuclear transfer. Okay, let's start with cuttings, and I want to do that by talking about bananas. Okay, so you and I all eat bananas. We love bananas. Bananas are great, and there was a commercial in the 1990s that put a song that is incessant in your head and is really hard to get rid of if you still remember it. Now, how do we cultivate bananas? Basically, how we grow bananas is quite simple. You grow a banana shoot, which looks like this. And then as it grows, it forms new shoots asexually from its base. You cut said shoot, and then you replant them in a row just about a meter down or 2 meters away, and then you rinse and repeat, and next thing you know, you have a banana plantation. Okay, now why do we do it this way? Basically, it's economical, it's efficient, it's fast, it's simple, and it maximizes the profits. You start with one plant becomes two becomes four, and next thing you know, you just have a whole plantation of them. And that's actually how most of the bananas you and I eat. Basically, almost every banana that you eat is genetically identical to every other banana that you see on the shelf. So the next time you go to a supermarket and you grab a banana, you can then brag to your mom about how that banana is genetically identical to the one that she grabs right next to it. Okay. Now, what's the downside? Well, let me introduce to you to the problem with cuttings, right? This is the Gros Michel, I think that's how you pronounce it, the Gros Michel bananas. Now, this was the dominant export strain prior to the 1950s, and they were cultivated asexually through the shoots that I mentioned earlier. The problem, however, was that somewhere along the lines, a fungus called the Panama disease, which originated, I think, in Panama, got to it, right? Now, you and I have never seen this banana because we don't eat this anymore, because the Panama disease has basically created the world's biggest banana blight and banana shortage in the world back in the 1960s or something like that, right? And they kind of went out into export. Now, in parts of Asia, you'll still see this banana around because that particular region or location didn't quite get infected with the Panama disease. And basically all that happens is, like, the banana could have a loose clump of dirt in it, or the shoot could have a loose clump of dirt in it, and then that's how the fungus just makes its way to new plantations. And then once it reaches a plantation, it completely devastates that particular plantation because all the bananas are genetically identical to one another. And so as a result of that, it just completely, just plummets the population, and then they all die because of the disease, right? It's pretty grotesque. It makes all your shoots go brown, and then the banana fruit themselves have this kind of black fungus growing inside of them, and it's pretty rotten, and it looks pretty gross. Okay, so basically that's how the Gros Michel went out of production. Now, instead, after the Gros Michel got hit by the Panama disease, we managed to find a new cultivar, or a new strain of bananas, which is called the Cavendish banana. Now, this banana looks a little bit more familiar because you and I buy this from the supermarkets all the time. They are Cavendish bananas, and basically, production of the Cavendish banana kind of became more popular, if you will. They were always around, but became more popular after the Gros Michel's demise, because primarily it is resistant to the Panama disease, right? On top of that, it also stays green for longer, which actually allows for a longer storage time. In fact, the banana is picked about 7 days before it is actually ripens, and then that's it's stored in the freezer or whatever, and then it's stored in a truck or whatever, and then it's taken to the shops, and then it actually turns yellow during that time. Now, about 47% of the world's export, including the majority of Australian banana exports, are actually Cavendish bananas. Okay, and so this is the new replacement, and we're so blessed because it is resistant to the Panama disease, right? Now, you would imagine between the 1950s and now, what is it that we have, oops, I don't know why that's gone backwards. What is it that we have learnt? Well, interestingly, Dan Keppel, the author of a book called Banana, the Fate of the Fruit That Changed the World, said this. He goes, every single banana scientist I spoke to, and that was quite a few, says it's not an if, it's a when. And 10 and 30 to 30 years, he said. It only takes a single clump of contaminated dirt, literally, to get this thing rampaging across the entire continent. Okay? Now, basically, we have learned absolutely nothing, right? Cavendish bananas are still cultivated by asexual methods and cuttings, and recently they have found that the Cavendish banana is susceptible to tropical race 4, which is a strain of the Panama disease, found on certain banana farms in Colombia, leading to a bit of a crisis in Colombia at the moment. And it is basically just a matter of time before it makes its way around the world. So, enjoy the banana that you are eating right now, because it's only a matter of time before it goes out of production because of the Panama disease. So that is a cutting. Now, a grafting is when you transfer parts of one plant onto another plant, right? And basically, you're going to have multiple parts of a plant growing on each other plants. And a way to think about it is when you have kind of like vineyards and grapes and stuff like that. So how it works, right, is when you remove a part of the plant at an angle, that's the thing that you remove is called the scion or the scion. I'm not quite sure how you pronounce that. Leaving behind just the rootstock, and then you're going to graft a new scion into the rootstock, and then you're going to wrap it and wax it so that the plant can fuse together and grow. So here's an apple tree with 2 strains of apples growing on the same branch, which is pretty cool. Now, the classic example that you and I are familiar with are grapes and vineyards. So vines often take a while to develop a strong and deep root system. As you know, grapes are vines, and so getting that strong root system often takes a long time. However, when you harvest the grape, you kind of have to kind of remove everything, right? And so basically new vines can instead be grafted onto established rootstocks. Now, that means that the new vines immediately have access to water and are therefore able to grow faster as a result of that. And so when you go to the vineyard, that little stem, that's not actually the trunk of the vine that's growing currently. That's actually an old vine that's just kind of been left there, and then it just kind of thickens over time, and then they graft the new one onto that, allowing them to get access to water, which is pretty remarkable. Okay. Tissue culture. Tissue culture is when you have fragments of tissues from plants or animals that are transferred into this kind of artificial environment, like a petri dish or something like that, which allows them to survive and reproduce, right? Theoretically, right, if you take a part of the body, like, a piece of your skin or a piece of tissue, and you keep feeding it the adequate nutritions and hormones and all that kind of stuff it needs to survive, the cells should theoretically be able to survive and grow, right? And that's what they actually can do, right? So, this is pretty remarkable in the sense that they will continue to divide via mitosis, and they will continue to grow so long as you keep giving them that nutrients. Now, there is a whole host of applications that are available as a result of this. The first of which is cell research. By actually reproducing that cell in an artificial environment, we can keep learning about it and understanding it and developing more breakthroughs through application of medicine, etc., and pharmacology, etc., right? You also have what we call tissue therapy. Tissue therapy is when you actually regrow that particular tissue or that particular part of the body in an artificial environment to put it and return it back to the individual. And a good example is a skin graft. So, for example, patients with a skin defect or a burn, for example, might have a portion of their skin taken off them. And then those particular cells might actually be then placed in a tissue culture and reproduce en masse, and then maybe a bit of collagen or some other materials are added to then turn that into a graft, and then that graft is then replaced back onto the skin, allowing it to then allowing it to then regrow over that patch of burned skin or whatever, and allow healing, which is pretty remarkable. And this is here are some images of microscopes of particular tissue cultures in different environments and stuff like that. Okay, so pretty remarkable. Another application which we often don't think about, but a bit of a scientific breakthrough, is actually cultured meat. Rather than hunting or cultivating livestock, you can just isolate a section of the tissue, particularly the muscles, and then you can actually give them the right environment to actually regrow, and then you can then package and eat that cultured meat, which is sounds pretty freaky at the start. It's like something out of Black Mirror or something, but pretty cool if you think about it, because it means that then you then don't need the animal. There's no animal cruelty and slaughtering, and then also you can probably do it in a sustainable manner, because as we are aware, growing cows take up a lot of water and carbon, and adds to the greenhouse effect and blah 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 blah, right? Now, another one is plant micropropagation. Now, it's called tissue culture, but you can really just think of it as a fancy version of it's just another fancy version of plant propagations and growing, where you grow the plant from its individual seeds, seedlings, etc., and you grab a part of the plant that can actually grow and act as a cutting or vegetative propagation, and then you place it in an environment where it has a lot of nutrients, like an agar gel or something like that, and you grow it for a period of time until it gets big enough that you can transfer it into a greenhouse or another location. Surprisingly, actually, high-end marijuana dealers actually grow their marijuana through tissue culture, because by refining or putting it in a controlled environment, you can refine its growth, and then that produces a finer quality of marijuana, which none of you should know about, because that's not something that you should dabble into. Okay. All right, embryo splitting. Embryo splitting is the artificial division of the embryo to produce multiple offsprings. Basically, it is artificial creation of twins. So you can imagine how twins are created when you have one embryo splitting into two, and then each of them getting their own little placenta and turning into an embryo, and then the baby. Embryo splitting is when we do it artificially, right? So basically, we take the embryo, and then we split the embryo as it's growing, and then we then isolate those particular clumps of cells, and then they turn into more embryos, and then we then put them into a surrogate mother to then breed or reproduce the cloned offspring, right? So, for example, here is the original sheep that you would have gotten your embryo from that was pregnant, right? And then what you do is you remove the clump of cells, and then you then split them into different clumps, and then you then let them grow in probably a tissue culture for a little bit, and then you then reinsert them into surrogate mothers, and then in the end they all produce the same sheep that is genetically identical to the original one here, despite being mothered by different surrogates. Okay. It is used for cloning particular strains of interest, ignoring its parental DNA, right? So, for example, if you have like a really prized bull that is known for a particular a competition or meet or whatever, then what you want is you really want to make sure that that particular DNA is not contaminated by the cows, and so you might do something like you mate the perfect couple, and then you split the embryos into surrogate mothers, and then that's kind of how you end up with more copies of your prized bull, right? Now, how it actually happens is pretty cool. They actually have this little vacuum thingy where they put the embryo on, and then what they do is they jab this little thing on it, and then it isolates those particular clumps of cells, and then what they will do is then they will take that, put it into a tissue culture or an agar plate, and then it regrows them into your individuals, okay? And this is largely also how they do a lot of IVF as well, okay? All right, here's the last one, and probably the most complex version, but probably the most remarkable version, is what we call somatic cell nuclear transfer, or simply nuclear transfer, where you produce a viable embryo by transferring the nucleus from a body cell into an egg cell. Now, this is kind of like, science frontier stuff. How it works is pretty remarkable. You take the nucleus with the genes you want from an existing somatic or an adult body cell, like a skin cell or a tissue cell. Then you take the egg and you remove the nucleus from another egg, right? From that egg, sorry. And then you fuse the nucleus of the cell you want with the egg that's now emptied of its nucleus using an electric shock, which sounds like something out of science fiction. And then you grow an artificial zygote into an embryo as a result. Now, if you were to look at this infograph, you can kind of see here. So for example, let's say this is the cell of interest and you want this particular DNA because of whatever reason, it's got some sort of particular genes you really want. And then what you do is you get rid of the nucleus, you remove the nucleus from the rest of the cell, you take an egg cell, which has all of the organelles that you want, you get rid of its nucleus, and so you now have the nucleus of one cell with the empty egg of another cell, and you're going to fuse it together with a little electric shock, and then that's going to then form your now new individual zygote that's been artificially created. Then you put it into a bit of a tissue culture, and you grow that into a larger clump. And if you are doing reproductive cloning, you might end up putting that in a surrogate mother so that it develops into an individual. If you are doing it for therapeutic cloning, which is where you, instead of growing into a new individual, you're only growing it for that tissue of interest, then you put it into tissue culture and you keep going until it has its particular application. Now, pretty remarkable if you think about it, right? Because what it means is you can replace cells that were previously thought to be irreplaceable, right? So for example, right, let's say you have an individual, and you are really interested in cloning that particular individual, like a mouse or a sheep or whatever, right? Then you can take its adult cell with the DNA, because remember, the DNA has all of the genes for everything in the individual, right? And if you take that nucleus, and if you can figure out to put it back into an egg, that's going to recreate into a clone that's going to develop into a new individual, which is genetically identical to the original adult, right? And so that can have applications in all sorts of places, like if you wanted to replace certain organs, if you wanted to replace certain genetics in the individuals, etc. You can just regrow a new one from its body cell, right? And then clone it and then have it turn into a new baby. And then, that then results in the availability of whatever parts that you need from that individual, right? Something right out of a sci-fi movie. On the other hand, let's say you just want to replace skin cells, right? So you take the skin cells, you apply the same thing, and then you put it into tissue culture where it then regrows into more skin cells, or something that is not so easily, reproducible, like for example, a nerve cell, which, as we know, nerve cells never really reproduce. Once they form, they stay with the individual for its entire lifetime. If you can take the nucleus of a nerve cell, put it into an egg, and then allow that to regrow, then you could possibly regrow that into more nerve cells, which then are then able to be replaced back into the individual's brain or parts of the body, and then that could cure diseases that would previously affect the nerves, like MND, ALS, etc., right? Pretty amazing if you think about its applications, right? So for both therapeutic cloning and reproductive cloning, there is extensive applications if it was possible as a result of current breakthroughs. Now, we can't really talk about cloning if we don't talk about Dolly the sheep, right? So Dolly the sheep was cloned first in 1996, and it was the first mammal to ever be cloned using this method of somatic cell nuclear transfer. Dolly lived for about 6 years and then was eventually euthanized because of lung disease and extensive arthritis, and gave birth to 6 healthy lambs. So by all means, she lived pretty much a full sheep life, if not, actually, maybe not a full sheep life, maybe about half of a sheep life. Now, Ian Wilmot, which is this guy here, the guy who kind of headed up the entire project, kind of describes the extraordinary, kind of story of Dolly by saying this. He goes, "Dolly seems a very ordinary sheep, yet as all the world acknowledge, she might reasonably claim to be the most extraordinary creature ever to be born. Dolly has one startling attribute that is forever unassailable. She was the first animal of any kind to be created from a cultured, differentiated cell taken from an adult. Thus, she confutes once and for all the notion, virtual dogma for 100 years, that once cells are committed to the task of adulthood, they cannot again be totipotent, right? And so based on our understanding of stem cells, you can see if you can regress the cell back into a totipotent state, just exactly how much application that could bring, ignoring the whole stem cell debate altogether by being able to clone this as well, right? So Dolly was done via that same method. You had the egg cell donated from a Scottish blackface ewe, whereas you had the nucleus donated from an adult Finn Dorset ewe, and then which are both female, by the way. And then what you got was the final product of the somatic cell nuclear transfer being in a surrogate mother producing Dolly, which is a Finn Dorset lamb, right? Completely different from both adults. Okay. So pretty remarkable story of how cloning can have, can come about. Now, we can't really talk about cloning without talking about its ethics. So if you want to think about kind of some of the problems of cloning, there were a few problems, right? So for example, there were a lot of attempts that are required in order to have a successful cloning. Dolly took 277 attempts, so you can imagine all of the failed attempts and what that means for individual cells. Developmental issues often occur in clones, so many die early in the developmental stage. Dolly only lived kind of half the general lifespan of a sheep. A lot of it comes down to the fact that the age of the sheep basically picks up from the age of the adult, right? So we talked about telomeres shortening. Well, if you take an adult cell, the telomeres have already shortened by a certain stage. And if you put it back into a newborn, and you create a clone out of that newborn, then, that newborn basically picks up the age of the adult. And so it's like a 12 or, a 6-year-old baby, as you will, right? Now, there are also, significant ethical issues, thinking about cloning as well, right? So, the applications involve being able to clone parts or whole organisms for products, right? And then you can regrow things like limbs and other vital organs in your clone and then transfer them back into your individual. And the opposition can really be thought of whether or not that interferes with nature or playing God. Should individuals be cloned? Should parts of an animal be cloned just to provide organs for another animal, or should that animal kind of have its own kind of animal rights and be able to live its own life, etc., etc.? And then if you talk about it for humans, the same thing, right? Is it okay to clone humans? Is it okay to take a person and then, create a new genetically identical one, for especially things like regrowth of limbs and body parts and, those kind of things, and lots of ethics surrounding that. So a lot for you guys to think about, and a lot of you guys to consider in terms of the ethics. Now, currently, cloning is not allowed, but if it were, what were some of the considerations you would think and whether you would be opposed or for it, given its advantages and disadvantages? Okay. All right, that's it from this video. I hope you guys enjoyed it. And that's actually it for this chapter as well. So I'll see you guys in the next chapter.